perspective. From my perspective, you guys are the best and the brightest. You're in a great environment, and I would most encourage you to figure out how to get yourself in a productive vein and think about what you want to accomplish. You've got the skills, you've got the environment. Think carefully what it is you want to do with yourself. One of the things I was told was, the science is nice, but all of the actual complicated problems involve people, and I have found that to be true. And so I think beyond that, a quick summary, I agree with if you're in graduate school, you're in a research group, get some skills, do really good, learn how to do something really well, partly because you can build from that outwards, and partly because it's just important to identify good work. And there's a process of doing good work that is eminently transferable across things. I did spectroscopy of myoglobin, and I wrote a Ph.D. thesis on myoglobin. Shortly after I graduated, a group at University of Texas knocked myoglobin out of a mouse and reported no effect. I got to the theoretical biology group at Los Alamos. It is mostly infectious disease, and so it's a mixture. The HIV database is there, so Betty Korber has actually handed off some of her vaccine designs for HIV to J&J, and it's been, a partnership and all of that. We're currently working with the Defense Department. USAMR, the Army, has actually brought many vaccines to phase 1 clinical trials because they're responsible for the full breadth of things that might be aerosolized and disseminated, for which there's no real market. And we're doing that. And we also do a lot of high performance computing because of the simulation and such for that weapon mission. As Jennifer sort of alluded to, we do have to think a lot about national security issues. And so it's a little dark, I won't go there, but I will say we've done a lot of epidemiology. We have to deal with a lot of problems that sort of fall through the cracks, one of which is global biosurveillance. Whose job is it to look and see what nasty diseases are showing up in Democratic Republic of the Congo, and whose job is it to articulate to Congress what kinds of global programs should actually be put in place? And so, coming back full circle, I've been deputy group leader and group leader since 2007, so I've spent almost 20 years doing management. I think one of the things that surprises me is how frequently group dynamics can actually be qualitatively improved. People tend to just start suspecting each other and fighting, and often it's for no reason at all. And so make sure when you're in a group of people, you learn how to interact with the group. When you learn how to, grab success from the jaws of failure. I think that's important. In terms of the technical, we did a lot of scenarios around epidemiology and disease planning for, what would happen if infectious disease X were deliberately aerosolized and disseminated. And there's a lot of theoretical biology. A lot of it, though, came down to disease progression. So we approached the VA a long time ago and said, hey, you guys update your database nightly. You have microbiology reports on pneumonias. You do MRSA swabs on every inpatient admission. And the response was kind of interesting. They said, for acute disease progression, the VA is not a great data set. You're probably better off with a specific study. But for chronic diseases like diabetes or behavioral health, you're not going to find a better database. It's phenomenal. And then serendipitously, Rick Perry and Secretaries Perry and Wilkie had the same thought, and they actually brought to the table and said, hey, let's bring the Department of Energy's high performance computing and AI to the electronic medical records. They have, I believe, never been shared with companies, and it's a beautiful resource. So that's sort of the prelude to all of this. I was told once I was disorganized, and I talked to a bunch of people, and I decided that no, I was discombobulated. We're working on a lot of different things. So this will be a little bit that way, but I think the way I would think about this, and I think the way I would think about the interdisciplinary science as a whole is, this is an example of what happens when you grab at an endpoint and work backwards to the science that's needed. And I would encourage people to think about that process as you think about careers. So again, the collaboration in red are the VA medical centers. These are all lists of PIs. Suma is the program manager at Alamos Research and Development. Amy Justice and I led this overall effort. And it's universities, national laboratories, and VA. And the core of this is built around a paper which is using AI to predict suicide risk in billions of veterans. So the VA has a mandate to reach out to high-risk potential suicides for interventions. And that's all published. It was published in January of this year. But I think what I really wanted to wrap around this is think about how the ideas come back all the way to multiscale biology. So don't stress the details. And then I'll look for how to advance the slides. There we go. So I'll start with a note about AI. Our mandate was to bring AI and high performance computing to these problems of the VA. When I started 5 years ago, I listed the pros before the cons. I think now I'm listing the cons first. There's a beautiful article by Gary Marcus, who's one of the founders of a lot of these AI techniques. And the question is, how much of this is hype and how much of it is real? And I think at the core, the AI is a tool that provides mappings, and you can do with it what you will. And the top 10 limitations are actually kind of damning. It's data hungry. You can't take the basic way AI works is you look at millions of ways to solve a problem, and then you can back calculate all of the complexities, et cetera. Unless you have millions of examples, I'm not sure how it differs from logistic regression. It's shallow, it doesn't transfer, it's not good with hierarchical structure, it's not good with open-ended problems, it's not transparent with features, it's not well integrated with prior knowledge, it can't distinguish causation versus correlations, it assumes a stable world, it can't be fully trusted, it's a pretty damning list. So the pros, what does it do well? I think there's two things at the end of the day. It's a really flexible tool set that creates nonlinear models. You're not building in assumptions of independence, you're not forcing a certain model onto things. And the second one, which is a little bit more subtle, but I think it's the enduring answer, it lends itself to transfer learning, where you do dimensionality reduction by training on one problem, and then you can take a much smaller data set to fine tune it for another one. So the example for that is ChatGPT was trained on predict the next word in a sentence. And then 99.99% of the parameters are frozen. And then you can train for domain specific or fine tune for domain specific. And so that's really, the visual at the bottom is you train all of your parameters on one problem, and then your output layer, you re-optimize or you fine tune for another problem. So that's, at the end of the day, that's the summary of how the AI got used. So turning our attention to the problem of predicting suicide, as I said, we were asked to do cardiovascular disease, suicide, and cancer, and so this is one. So, the subtitle here, there's a lot going on. And I think it's like so many of the problems that end up at the national labs, it's complicated because of all of the different aspects to it. So at the individual level, you have a lot of drivers. You might have behavioral health issues, you might have addiction issues, you might have family history, you might have circumstances. But that sits within relationships, community, and society. And I think one of the reasons it's important is societal, you have to make decisions about spending resources and such. But I think for this audience, there is also a strong heritability to this stuff, and which means at some level it's a bioengineering problem, it's a biochemistry problem, it's a genetics problem. And so the real question is, how do you tease apart the different aspects of the problem so that you can move forward? And this slide is Eric. So if you look at the VA data, and the VA said this was important, and it is. It's probably a third of their healthcare budget. But I think the broader point here, VA data and researchers are just a unique and world-class resource. So if you think about data-driven science, and you think about how big the data sets are that you look at, the VA has had an electronic medical record since 2000, and it's all inclusive. You get the pharmacy polls, you get the laboratory results, you get the clinical notes, you get the radiology reports, you get, so anyway, so this is just a statement of the size of it. Again, 25 million patients, that's 1.4 billion appointments, 7.7 billion laboratory results. So this is ways that you can conceptually, you can characterize the diseases, you can characterize the treatments in terms of pharmacy, clinical orders, surgeries, et cetera. And then there's 3 domains that I'll come back to at the end. And I think this is really where you can come back to the bioengineering and the multiscale biology. You've got 200 million radiology reports and associated images, which are also often 3-dimensional and multimodal, like functional MRIs, each one of which has a radiology report with some kind of a ground truth associated. You've got 3.2 billion clinical notes, and a lot of the questions about disease severity or other issues that are going on, reasons for discontinuing treatment, Those are all captured as unstructured data in clinical notes, and you've got to figure out how to pull them out. And then finally, the Million Veteran Program is a consented study which has over 1 million people enrolled. One of the first things they did was take a blood sample and do a genomic profile. So measure 750,000 SNPs, and that got imputed to 50 million SNPs. And the point is you can connect it back to the clinical record. And so I think these are the three components that really come back to the bioengineering is, can you really figure out how to mine that data and how to formulate the problem? There's really three types of questions that are relevant, and in broad sense, patient screening, can you identify the at-risk patients? Risk factors, can you identify the biological drivers of disease? And then the hard one, which is in red, is comparative effectiveness analysis, which is, can you tell what would happen to this patient if they did insulin or they didn't do insulin? And I don't want to say that's in the too hard to do box, but it's astonishingly difficult to correct us for all of the biases. And I would also, part of the reason I'm going through all of this with you guys is this is similar to the problem of developing new biomarkers and developing new measurements, is correcting for all of the biases. And I think if what you guys are doing is anything like what I was doing when I was in school, the samples are what you can find. And what I want to throw out here and plant in the seed, because I'm anticipating you guys will continue working for another 50 years, is that you really should think about the clinical study design and really think about, if you're going to attack things like mental health, how do you harness together the higher level scales of the patient care with the sample collection and experimental design? In other words, we saw the mass spec, you could do things at high throughput. Well, hey, let's put samples that are chosen from the clinical records of the Alzheimer's disease and not necessarily from smaller things. So again, starting at the end point, this is slides. This actually comes from the Institute for Health Metrics and Evaluation. It was funded by the Gates Foundation pretty heavily to prioritize global health spending. And what it is a representation. On the left is years lost to disability, and on the right, it's death. And so the area of these squares is proportional to its contribution to morbidity on the left and mortality on the right. And it's actually surprisingly stable. This is a global average. The quarter of the world that's less developed has a much higher representation of infectious disease, but other than that, it's pretty stable. And my point is that the outcome definition when you do this is absolutely critical. And so if you think about mental health, behavioral health, it's in the red square here. It's actually 15% of years lost to disability. And they put a lot of work into thinking about this, all of this to be measured. But the point is, it's bigger than cancer. It's as big as cardiovascular disease. It's a huge problem. And I think all of us anecdotally know this. If you look at deaths, on the other hand, you won't see the mental health, behavioral health issues showing up in death. About 1% of it is self-harm. But if you look at the causes of mental health, it's depression, anxiety, schizophrenia, autism, etc. So again, all the AI in the world doesn't help you if you formulate the question inappropriately. And I think that gets glossed over, and one needs to think about how exactly to do it. I'll just stop. So if you look at the VA database, this is a histogram of first suicide attempts. This is the monthly incidence across the VA Medical Center. There's 4.2 million active patients today. There's 25 million patients who have touched the system. So green is first reported suicide attempt. Blue is first reported overdose. Red is suicide. So I will just tell you as an outcome, if you have 43,000 suicides, that's not sufficient to do a statistical model to constrain all of the drivers. Your model will end up pulling out the indicators of suicide and not necessarily the mechanism. On the other hand, if you construct a binary outcome, which is if there's a death by suicide, then that's the outcome. If there's not a death by suicide, then first suicide attempt, otherwise first overdose, that gives you 250,000 cases. And that is enough to constrain a statistical model. The problem is there's no principled way of going from complex outcome case control study to prospective study. This is where the AI really helps. And that's the core of the technical story. So in some ways, what I'm telling you is Biostats 101. In some ways, it's a little bit more subtle than that. I guess I would ask a question of how many people in the room have actually thought hard about a logistic regression problem? So there's a couple. In some ways, it's an astonishingly simple thing, but in other ways, it's a little bit subtle. If you have millions of patients, you could, so a logistic regression is just asking the question, if I have a whole bunch of predictor variables, blood pressure, previous suicide attempt, hemoglobin A1C level, et cetera, can I construct a model which predicts the log of the odds of the outcome as the sum of independent contributors? And so this is the definition of a linear risk factor. This is where saying smoking adds 20% to your risk of a heart attack or whatever. And the point is, with millions of patients, you can actually constrain 200 time-dependent variables if you have 250,000 cases in that. And that has to sort of be the starting point and the foundation. And then once you have that done, you can do more fancy things like think about the AI. And then the other key component of it is model selection. So when you're optimizing the least squares, you also have some sort of penalty for having a more complicated model. And so that's at the core of what we're doing with statistical analysis, is trying to predict an outcome, start from a logistic regression and find the risk factors that correlate, assuming they're independent, and then penalize for model selection. So if you do that for suicide, these are the binary variables. All of these are categorical 0-1 variables, so you can compare the coefficients directly on a scale. And again, you can see the logistic regression and the different shaded colors are whether it's 3 months proximal to the event or 7 years distal, 7 years earlier, a little bit more chronic. And so you can see diagnoses, treatments, indicators, demographics, and biomarkers. And there's a lot of interesting things going on here. I think that the purpose of this is not so much to dive into the details, but the broad point is with the VA data and millions of cases, you can actually look across a broad range of things. I will tell you right away, there's a couple of things. One of them is the most useful thing you can do is just ask somebody if they're suicidal. That's a PHQ-2, PHQ-9, and it's strongly predictive. But you can also see things like hypoxia and vascular damage are independent biomarkers that predict suicide. Those are actually useful insights. If you're looking in terms of medications, the problem is the coefficient that they're preferentially prescribed to the sicker people. So you have to think carefully to interpret. Okay, so what do you do with these models? So when you train a logistic regression model, you can turn around and use that model to predict, for example, 2-year probability of either suicide-related behaviors on the left or suicide on the right. And you can evaluate. So what we do here is we stratify the patients by the score. So these are people who have a high score. They're predicted to have a 90% probability of an event in the next 2 years. And these are the people predicted to have a 0.1%. And then you just take all of the patients in given subgroups, so schizophrenics are in magenta, bipolar is in cyan. And you just say, this group of patients was predicted to have this score, and then you go back and you look 2 years later, what fraction of those patients actually had the event. So if your model is well calibrated in all the subgroups, the dots are on the line, and this is not the linear model, this is the AI model, and it's in fact well calibrated in all of these subgroups, in the sense that the patients with the given who are predicted to have a 50% probability, in fact, half of that group had an event. And you can see that the model actually works well in all of these subgroups. So this is subgroup analysis and calibration analysis. Everyone always complains about the interpretability of the AI, but I would sort of throw out there that something like this, model calibration in relevant subgroups, is actually a pretty stringent test of how things are going. What you get out of that is the risk histogram at the bottom, where you can see the schizophrenics have 2 log units higher risk than non-schizophrenics, and that's a univariate comparison. And that works out to a factor of about 8 risk. So you can start to decompose a lot of different things. You can start to see the different subgroups, and you can start to play and predict with this model. That's fine. I'm missing a slide, but that's okay. So I was going to go through from this point some of the complexities of the model. And I'm not sure what happened to model complexity 1, so I'll start with model complexity 2. So one of the problems with observational data is that you have what's called a confounding effect, which is some of your predictor variables directly impact the outcome. And there's very little that you can do about this in terms of study design. This is why randomized controlled trials are done. Whether they effectively solve the problem is a question. So the problem is that patients with frequent healthcare visits, they're more likely to have their suicide attempts and overdoses reported when they occur. So if your outcome variable is a suicide attempt, then just the fact that you're going to the doctor a lot is going to increase the odds that it's identified. So that's called confounding. And so what it's related to the statement somebody once said that if you wanted to decrease the prevalence of COVID, you just test less frequently. It's that exact idea. So the problem with it, and the reason I bring it up, is if you look at now at the calibration curve for this, now instead of subgroups based on mental health diagnoses and treatments, you have subgroups based on how frequently you're utilizing the healthcare system. You can see two things that are important. One of them is the effect of healthcare utilization is actually way bigger than the effects from the schizophrenia and the bipolar, which is saying you need to actually correct accurately for the confounded behavior. And if you look, the model is actually well calibrated for the people who go a lot and the people who go fairly rarely. And so that's one of the things the AI did really well. The other thing you'll notice is that on the left is the model for suicide-related behaviors, on the right is the model for suicides. The reason I'm bringing that up is suicides do not get taken to the hospital. They're reported through the CDC and the National Death Index, and that actually is not a confounded observation. And so even though the suicides are a lot noisier in the risk histogram, you can already see that the bias or the predictive ability of number of visits is much weaker among suicides than suicide attempts. So it's a way of getting a handle on the confound. In terms of how you, I'll move on. I don't want to get too drowned. Need for study design. And this is, again, something that much of biostats is dealing with. And to a certain extent, you do study designs anytime you do a study in your laboratory. What I'm really proposing is that you sort of try and do your study design more carefully with the patient behavior. What I've shown here is, in some ways, it's food for thought. And it's a little bit of a statement of this is what the real world is like. So if you take one of the patients who was at high risk for suicide-related behaviors. This is a single patient. The x-axis goes from 2000 until the patient's death in 2019. Every circle on here is an observation. So from here up to here, these are pharmacy pickups of medications. So the diabetes drugs are here. And then here's the antipsychotics, the analgesics, sedatives, the anticonvulsants, the antidepressants. The point is there's a lot going on here. And reducing things to a binary variable is a little bit problematic, and there's a lot of implicit assumptions and decisions that are made in the course of the trial. I don't think when people do randomized clinical studies of particular medications, they use patients that are taking a dozen medications. But the fact of the matter is that's how they're used in the real world in many, many cases. And so when you think about the study designs, there's really two choices that are relevant here. You could take an event like a suicide attempt, the red line here, and you can look backwards from that, and that's your case. And then you can somehow select a matched control that you want to compare to who didn't have an event. So that's a case control study design. And that is one way, depending on how you choose your controls, that you can make the confounding variables cancel out, effects of confounding variables cancel out. The prospective screen is to just pick a calendar date, predict 2-year risk forward. And they're both relevant things to do. But I think it's just important to look at the complexity of the real world. That's not to say every VA patient has this complex of a medical history. It's to say that this is part of this. So this is, again, the two types of study designs. So the left is a retrospective case study. This is what you're using when you want to maximize the statistical power and you want to control for or match on things like healthcare utilization age. And you can see the statistical power. There's 250,000 events if you use a combined outcome and you use a case control study design. And if you insist on training prospectively with suicide as an outcome, you can see the statistical power is down by close to a factor. So this leads to the third model complexity, which is that your variable selection. Remember, I said at the very beginning, the model selection, the lasso model selection sort of automatically helps avoid the overfitting by just throwing out variables until you have sufficient statistical reasons for including them. Well, the problem is that your variable selection procedure is going to depend on the statistical power of your study design. So if you have a model of suicides and a model of suicide attempts, or a model of suicides in male veterans versus female veterans, they differ enormously in the statistical powers. You get different results out of your study design just for that reason. So it makes it very hard in a straightforward way to compare those two statistical models. And so what I'm showing here is just comparing the coefficients in a model for the retrospective case control study and vertical the model for the prospective study. And again, I don't want to go into the details here, but the point is the higher statistical power model uses more variables and it gets back at the mechanism. The take-home message from this, which again, what you can do with AI and the transfer learning is you can train the bulk of the model on the high statistical power, and then you can fine-tune it for the suicide. And what that allows you to do is it gives you a better comparison of how do the two different things compare. And that's something that you can't do with computers. So again, this is arguably not worth running through, but I'll say it. So what we used was something called ensemble transfer learning. And the idea is you create an ensemble of models that are all predicting, they're all fine-tuned to predict the prospective suicide risk. And you can see retrospective case control, logistic regression, neural nets, random forest, CADNet, Cox models, et cetera. You solve the problem in a variety of ways, and they all have different biases, and they make different assumptions. And then you use this as an ensemble of models together with the important variables that you want to fine tune on, like gender, age, et cetera. And you have a second stage of the fine tuning, which is a much lower dimensionality, which you can do on the rarer events. And you could create an ensemble model for suicides and for the suicide attempts. And I think this is very generically going to be a useful set of tools where the AI can shine on that. I won't dive into details. This is actually published. Jumping to the implications of doing things like this in practice, this is just scoring different ways of how well did the model work. And one way of looking at it, if you're doing screens for suicide, you're really interested in the high risk end of the spectrum. So the VA has a program called ReachVet that every month the head of psychiatry of each VA is given a list of the top 0.1% of suicide risk. And the question is, by what factor can you enrich that population of people who go on to have to die by suicide? This isn't the combined number. And the answer is that number is somewhere around 6 to 8 to 10. It's depending on how you do it, the actual concentration of risk and everything, the actual ability to enrich in the top 1%. And I will say that with the AI, you have to be excruciatingly careful at making sure that you're not getting spurious increases in performance that aren't actually helpful. And that really people always say, oh, my model's not overfit. So what I've learned is you just ask them, how much does your model generalize? Can you take it to a different population? And it's surprisingly difficult to detect the overfitting. I will say just simply does the model work 2 years later? Already detects a lot of lack of generalizability that wasn't picked up by overfitting, really should have been picked up by overfitting. So AI, it is incredibly good at identifying artifacts, and I think doing everything you can to understand the generalizability. The second implication of the study in practice is the characterization of risk factors. So again, this is now looking at the relative risk for suicide and the relative risk for suicide attempts, broken out by the type of data. So the magenta are actually mental health diagnoses. And what's actually kind of reassuring about all of this is the mental health diagnoses actually do have the same ability to predict suicides and suicide attempts. And so they're actually, in a very real sense, not confounded. If you look at things like missingness of data, those are much more prevalent in suicides than suicide attempts. And that's, again, this is giving you a measure of the size of the confounding effect, et cetera. And again, this is another advantage of the high dimensional data sets, is you can make these plots and start to explore why your model is messing up in a way that you can't do if you just have 12 variables, et cetera, et cetera. So connecting back to the multiscale biology, again, this is what I said at the beginning. I think the real benefit of that is going to be to start to dive into the radiology data, the clinical notes, and the genetics, and especially put them in combination. And again, one of the motivation for the VA is a comparative effectiveness analysis. They really want to know, is aggressive hyperglycemic control beneficial to the patient or not? When they did the randomized study, it actually didn't turn out as many people expected. There was more mortality in the aggressive control. I think in terms of the mental health, it should be pretty clear. Harrison's Internal Medicine has prescribing guidelines for mental health drugs, which say, ask the patient what they've taken before and whether or not it worked, not making this up. There's lots of problematic issues. One of the difficulties in doing the comparative effectiveness analysis is the genetics is so critically important to it. Lithium works as a mood stabilizer for about 1/3 of the patients, and it's got very few side effects, but it's not prescribed because 2/3 of the time it doesn't work, and you only have one chance to make a first impression. So if you can dive into the clinical notes and find out reasons for discontinuation of treatments, is it that it didn't work? Is it that it had side effects? Is it that other unforeseen effects came out? Then when you go and formulate the question for the genetics, you can have a much better chance of seeing signals. And then similarly with the radiology. So if we do the genetics, and again, this has to be done with careful study designs, case control study designs. I will say if you do a genome-wide association study on the Million Veteran Program data, one of the top hits is dopamine receptor D2. As mutations in front of that are actually strongly correlated with suicide. That's the target of the antipsychotic medication, and it's known to be a important structure. So that's an example of a success. For radiology, it's tricky because the data are not all co-localized, but we did do studies with chest x-rays. We just took 200,000 chest x-rays from Boston and tried to predict edema, large heart, pneumonia, pleural effusion. And you can do this, it works. And there's a variety of use cases you can do with that. Incidental screening, biomarker development, et cetera, et cetera. And then the natural language processing, we played a lot with it for the suicide prevention. And there's just certain things that we know are important that are hard to find in the structured data, like job instability, housing instability, et cetera, et cetera. I will say we've been wrestling with, there's a lot of non-natural language in the clinical notes. There's a lot of cutting and pasting of templates. And the first time we did all of this, you could develop great models that predict with the NLP, but they're redundant with the structural data. So when you bring them together with the structured data, they don't add value. And so this comes back to the study design and how do you train the model and how do you think about the problem so it actually adds value. And then I'll close with this. I didn't get a chance to do a really good job on this. If you come back to the question of the bioengineering and the biological markers, you want to find a place where you can grab a hold and really play off the top-down approach against the bottom-up. And I would throw out to people, schizophrenia is probably one of the places where it's possible to do that. Depression is just simply too vague. There's almost certainly a lot of different underlying physiological things going on. Schizophrenia is relatively well defined, and it's kind of interesting here. This is a paper from 1983. Schizophrenia is caused by a fault in programmed synaptic elimination during adolescence. It's a hypothesis. This is from 1980. It was revisited in 1994. And again, the basic hypothesis is that when you have synaptic pruning from age 16 to 25, that too many things get pruned back, and this is the underlying driver of schizophrenia. And it's a very crisp biological mechanism. It suggests lots and lots of experiments. And the fundamental point of this is the recent paper, which is 2023, is this is essentially what's going on, is that you prune too many synapses, it leads to the dopamine not being effective in the frontal cortex, which leads to cognitive deficits and depression. This dopamine migrates to other parts of the brain, and when it gets to the audio cortex, it causes voices, and the visual cortex causes hallucinations. If you put in an antipsychotic that blocks the dopamine receptor, it blocks the hallucinations, and it doesn't do squat for the negative symptoms, the cognitive deficit. My point is that you can actually connect the dots in this case. And again, I think it's important to try. So there you go. Science is a team sport. Think carefully about where you want to go with your talent skills. Healthcare is in need of bottom-up approaches. I think the data access is complex, and I think people need to go there. And when I say complex, I'm sort of understating it. It's probably you that has to do all the work. But again, I would encourage you, multiscale extends from molecular all the way to medical and epidemiology. If you want real world data, you have to look in the real world. And then the other thing I will throw out there, genomics and evolution touch all the scales. You can't just interpret your evolutionary analysis in terms of the biochemistry and the cellular biology. Anyone who wonders whether or not mental health is heritable should buy a couple of cats and watch them. The behavioral phenotypes are very real. And many thanks for VA and all of these collaborations. So I'm happy to answer questions, and thank you for your attention, and good luck to all of you wherever you go. Yeah.
So your outcome variable here is binary, right? It's either there's a suicide attempt or not. Did you have any trouble using that as your outcome, because your AI model is kind of predicting a probability, and there might be a lot of cases where there's no suicide attempt for an individual that's very close to that, just because your ground truth is binary. And would you ever try to supplement that ground truth by using one of the confounding variables, either the PHQ surveys or kind of a mental health scale or something like that?
Let me just say an answer to your first question. Whether this was an actual suicide or not, I don't know. I mean, it clearly wasn't recorded as such. So you have to think of this from a statistical perspective. You make a choice on your definition of outcome. And the reason we did the combined outcome was exactly this. Probably half of the suicides are not recorded as such. There's a lot of literature about all of the problems you can get into by conflating common, less serious outcomes with rarer, serious ones. I think most of those problems are associated with the linear models and other choices that are made in the study design. So you have to proceed. This is the reason we had the combined outcome. Part of the reason was just statistics, but the main one is that we know the outcome is underreported, and we wanted to collect some. As far as what to do about it, I mean, you have to choose an outcome variable, do the best you can, and go with it. This came from a lot of discussions. The ReachVet program has been operational for 5 years, so we certainly worked with the operational people about how to do this and how to structure it. So it's imperfect, but I mean, that's the point of statistical models, is you have to work with it.
A different experience that there's many that maybe to identify like the inflection where it looks like there's a threshold patient goes past these particular suites of biomarkers, these higher values in their labs. Now we can say that there's some kind of higher risk as, and then use that as like the interventional point.
You know, it's kind of interesting, because if you think of it in a way that I think ends up being inappropriate, you can say, look, all you did by stratifying on the top 0.1% is enrich your cases by a factor of 6 to 10. That sounds pathetic. On the other hand, I think you have to look at it from a broader perspective. Anybody that's attempting suicide is almost certainly has a lot of underlying issues going on. That's an incorrect statement. I think so the short answer is no, there's nothing obvious that's happening. This is a really heterogeneous outcome variable. There's lots and lots of different drivers. And I think the part of it is targeting the mental health care. I think in terms of targeting mental health care, you have to recognize that the suicide-related behaviors and the risk factors for it in and of themselves can benefit from attention and treatment. And I think if you try to justify the intervention solely on that single outcome, it's going to be a real problem. So the short answer is no, there's no obvious inflection point. There's still a huge amount of randomness going on in predicting. On the other hand, when you broaden it out to look at suicide related behaviors, you have a much better ability to identify at-risk patients. But then the question shifts to, can you identify patients for whom a particular intervention will be helpful? And can you quantify that? And that's, we have not gotten to that. Does that answer your question? I'm sorry.
Yeah, thanks, great talk. There must be some shortcomings of the VA data set that you wish, you know, that you had more information on, say on younger subjects, or I imagine most of, more than half of the records are for males and things like that.
It's 90, it's somewhere between 90 and 95%. So that's for sure. There's plenty of shortcomings, but again, we're not going to get what we want. What's clear is amazing. So we just sort of look at it the way around. In terms of what we wish we had, I will say, I was going to say this before, I just forgot. There are so many problems that are multifaceted like this that involve policy, sociology, pharmaceuticals, etc. It is such a painful process to co-localize data across agencies, across industry. I mean, I wish the data sets could be co-localized. Our VA partners spent 3 years trying to get Center for Medicaid, Medicare data co-localized with the VA data. They had a data use agreement in place between the VA and CMS. That didn't extend to the Department of Energy in place. So we didn't get the CMS data, which means everything over the age of 65 is not as good as it could be. So yeah, I think if I had to pick one thing, it's the data sharing. And of course, everyone understands the need for privacy. I mean, these records are just, it's a hard problem. I'll just put it that way. I mean, the pharmaceutical industry, of course, is sitting on a huge amount of data in terms of all of the drugs that don't make it. How you would integrate that if you had access to it, I don't know. Because again, and this is where the VA is kind of interesting, because yes, it's a central database, but it's essentially 150 VA medical centers, each deciding how they want to put things into the database. And so it's got a lot of real world evidence. But anyway, stuff.
We talk about bottom-up approaches. I'm just curious to expand on that a little bit, because I think ideally, you know, preventative medicine, prevention care, patient care, whatever. We always think of it in 3 bends, right? You have the patient preference, the patient wants, which is backed by clinical expertise and what the evidence in the literature says. And so one of the most profound things that I keep kind of coming back to sometimes is that you don't need to know a mechanism to know that something is beneficial. And especially in a complicated case as suicide prevention. How do you envision bottom-up approaches working in an actual integrated healthcare center that's aware and cognizant of these issues, you know, things that work for certain different demographics?
It's somewhat, so there's 2 answers to that. I think the bottom-up approach sometimes fundamentally has to come from academia and industry, because it's a huge endeavor. And so this is one example of what I call a bottom-up mechanism. We have a mechanistic hypothesis. I think the schizophrenia is driven by the synaptic pruning, the excess dopamine, and all of that. That creates a slew of experiments that you could do to check for that. And so that's really what I mean by the bottom-up. In terms of why the top-down is so complicated, it's excruciating. So just think back to things that you've seen yourself. When people came out with the statins, they had the treatment guidelines that were based on reducing cholesterol levels. And then they had 10 years of experience and they discovered that, oh shit, that wasn't a good biomarker for this case. And when we looked at the actual outcomes, we had the wrong people taking statins. Hormone replacement therapy was the same sort of thing. It's excruciatingly difficult to correct for all of the biases that are in the observational data. And the real world data is missing some of the pertinent effects. You can't pretend that people don't take alcohol with their medications. You can't pretend that they're not on 6 medications. There's a case where we were looking at with receptor blockers and the blood pressure meds for the diabetes. One of them is more tolerant of missed doses than the other. And so in the real world use, they had very different impacts in terms of all-cause mortality. And you have to do an observational study to see that. You're not going to see that. And back to the VA diabetes study, they did a randomized controlled trial of aggressive hyperglycemic control versus not. 5 years in, there was no difference in strokes and heart attacks between the 2 groups, and the aggressive control had hypoglycemic episodes. 10 years in, you saw a difference. So it's the mechanism that's telling you you need to control a blood trigger, you need to look at insulin. And this is back to why I'm harping on the problem formulation. So what I'm saying is there's so many pitfalls in doing retrospective studies. Yes, we need to get better at doing them, and AI is part of that, data access is part of that. But you also need to come in with actual hypotheses that will reduce the dimensionality of the problem. And then, of course, the genetics, the radiology, and the natural language process have the possibility to do discovery science. You can find things. So I don't know if I answered.
I'd like to know, like, as you mentioned, there are lots of redundant data in this area, like, and you were using radiology reports, appointment notes, and everything. How did you choose, like, which one is better? Was it based on literature or
So at some level, we started with a set of hand-curated variables from the REACH-VET program. So complicated diabetes, uncomplicated diabetes. They're not the raw ICD codes, they're starting with that. But I will also tell you the lasso model selection did not throw very much stuff out when you had 250,000 cases. And so when you actually pull on the thread a little bit, there's a difference between a hemoglobin A1C level of 9, a diagnosis of diabetes, and a prescription for insulin. The prescription for insulin means they're treating it with insulin. The diagnosis means that they're actually thinking about getting the treatment done. The lab value is a lab value. They may or may not have done anything for it. So even though they're redundant pieces of information, they are subtly different things. And the subtleties have a lot to do with healthcare utilization and age. I mean, the slide that disappeared was the one that was looking at the age dependence of the risk factors. There is nobody over the age of 60 who's getting inpatient hospitalization for mental health. They've learned by then that they don't want to be inpatient mental health patients. On the other hand, overdoses go up nicely to compensate. And so there's what you think the variable means and how it's labeled. But then when you start putting in what looks like redundant information, you find out that there are actually subtle differences. And this is where the interpretation gets complicated. The other thing I didn't mention is there are no diagnosis codes for suicidal ideation before 2006. They simply didn't report it. And so there's homelessness diagnoses went up 5-fold during the Obama administration because they started looking for it. So so the idea is to put in all of the redundant variables because they're actually giving you different information. And then this is what the AI can start to do, is to disentangle this in terms of age and healthcare utilization patterns and behaviors that aren't reported. Does that help?
With that, I'd like to thank Dr. McMahon for we have a get to present to you to thank you so much for and with that, I think we can commence with our wonderful poster session. Come see the trainees posters, talk to them about their research, see what they're up to. I believe we'll have snacks and beverages provided, so there's that to look forward to as well. We'll be having it in the room next door where we had our lunch earlier. Thank you.
Maybe, oh, is it fun?
Yeah.
Yeah, that was.